Hello and welcome to today's Aquavitae meeting webinar with the Low Trophic Life series. Today we'll have Thomas Juhasz who will be talking about biofluorescence and as a tool to identify subclinical stress in low trophic aquaculture species. First of all, I just want to talk to you briefly about Aquavitae. Aquavitae aims to increase aquaculture production in and around the Atlantic Ocean by developing new species, processes, and products. The focus of the product is placed on low trophic species such as algae, sea urchins, and shellfish. You can read a lot more about Aquavitae in the link I'll be posting in the chat right now, or by following us on Facebook, Twitter, Instagram, LinkedIn, or YouTube. But for today's meeting, Thomas, before you begin, why biofluorescence? Why did you become interested in that?
Casper, I finished, I was an adult student in veterinary school. I finished in 2021 in Budapest, Hungary, where I'm from. And I was fascinated by the fish and invertebrates, while most of my classmates were with horses and dogs and cats. And one of the things that I noticed was that there really isn't a lot of diagnostic tools available for these types of animals before severe clinical signs manifest in them. So, and one of the ones that they would use was dissection, which I felt was not the best, diagnostic tool that can be available. And so I was watching a webinar about coral banding disease, I believe it was called, in the Florida Keys. And what they found is that corals themselves are biofluorescent, but in response to stress or losing their zooxanthellae, their fluorescence levels change. And so they began to start in the mid-2010s or so to actually assess the stress levels of reefs. And when other people asked, does this work in other species? Overall, they said, oh, no. But I thought, I think there's something to this because, around 2014, 2015, a slew of papers on different types of reef fishes and even sea turtles, started to come out. And that's a bit of my background of interest.
That's a very good start. Do you want to go ahead with your presentation?
Yeah, I'll share my screen. Okay, is everything good on your end?
Yeah, it looks good.
Okay, so hi, my name is Thomas Juhasz. I am based at the University College Cork in Cork, Ireland, and at Bantry Marine Research Station down a bit south from there in Bantry, Ireland. And I am a Marie Curie Ph.D. student, and I am looking at biofluorescence as a tool to identify subclinical stress in low trophic aquaculture species. And so just a bit about me. I was previously a biological consultant before I went back to school. Became a licensed veterinarian, beginning to specialize in aquatic species in 2021. And my interests are both fresh and saltwater species. The goals of my PhD are to determine whether biofluorescence plays a role in the health and welfare of species utilized for aquaculture, and to develop a non-invasive method to determine subclinical stress levels. Now, what is biofluorescence? The technical definition is the absorption of electromagnetic radiation, known as light, by a fluorescent protein or metabolite at one wavelength, followed by its re-emission at a lower energy and a longer wavelength by a living organism. And some of the organisms that I've been working with include lumpfish and European black sea cucumbers. You can see here on some of my photographs, on the left is a newly mature female lumpfish where only the tubercles are biofluorescent. A subdominant mature male lumpfish with very bright tubercles and I would call it a pretty dim, diffuse red biofluorescence. And in the European black sea cucumber on the right, you can see that the animal itself does not biofluoresce, but when disturbed, it releases a very powerful biofluorescent exudate. Now, why bother with biofluorescence? Now, biofluorescence potentially can be used in real-world applications to identify stress before clinical signs, reduce mortalities, and identify external parasites. So this is something that I believe can be used to identify what's going on in animals well before, crisis points or to the peak of the stress curve occurs. So when you have a peak curve in veterinary science, once the animal reaches that physiologically and behaviorally, it either has two choices: it either has to adapt or physiologically collapse, and that's when, that's when aggression and secondary infections like Pseudomonas or Pasteurella, Escherichia coli, et cetera, come in and do real damage. Now, the species I'm working with for my PhD, here are some of the pictures from Ireland. I've been working with two species of sea urchins, the green sea urchin, Strongylocentrotus drobachiensis, here with Phil James at Aquavitae, and also the purple sea urchin which is Paracentrotus lividus, the European black sea cucumber, the Holothuria forscali, lumpfish, Cyclopteros lumpus, excuse me, my Latin sometimes gets to me, and then Ballon wrasse, which Labris something, pardon me. Other species that I've worked with here aside from my primary focus, which is echinoderms, here in Tromsø, Norway, have been king crabs, sea lice, and seaweed. So I've had quite a few different things and species to work with. The methods: first of all, you would take your IMTA organisms and evaluate them both in regular light and excitation lighting. Excitation lighting is two types. You have UV light, which is under 400 nanometers, and you have it's called royal blue lighting, which has an excitation peak around 450 nanometers. 400 nanometers is where visible light begins for people or human vision. Once it is under excitation lighting, the data is recorded with a DSLR RGB camera fitted with a yellow barrier filter to take out any of the excitation lighting and to see secondary fluorescence, and as well as a hyperspectral camera, which is here at NOFEMA has been a VNIR ladder scanner, which has been very effective. The data is then subsequently analyzed using Breeze by Predictora for fluorescence levels and changes in those fluorescence levels. And on the right here, you can see like a diagram of the photography of the sea cucumbers in Ireland with a snapshot hyperspectral camera. Now, the difference between hyperspectral and regular or RGB cameras is that RGB has three primary color patterns that are recognized: red, green, and blue, while hyperspectral imaging takes over 1,000 different points of color. So it gives you a full picture of what's going on with an organism that you might not be able to see with a regular camera. Some early results. Here you can see on the top, this is a lumpfish that I reared in Ireland from a juvenile. And this was in a tank with 5 other like physiologically mature males. And this was the only male that produced this level of fluorescence. Everyone else was nowhere near that color, but they were the same age, general same weight and class size. And below is a European black sea cucumber with in the regular light, you can't see much, but under royal blue excitation lighting, you can see that the exudate that it is giving off in response to my handling is extremely fluorescent and very easily seen. So with Phil and the team, we worked on sea urchins. Now, the methodology was to take 3 types of stress-inducing factors and test the animals to see their responses. One was out of water under wet burlap bags to simulate shipping patterns for aquaculture. Cold water, which is the normal habitat of the animals, and as well as hot water, which is approximately 10 to 15 degrees Celsius higher to visualize whether any stress was occurring. There were two types of examinations in full spectrum vision. One was with a fluorospectrometer of the coelomic fluid of the urchins, which was about 2 milliliters taken from each animal, and also external scans of the animals themselves. Here you can see some pictures on the top of urchins in normal white lighting. In the middle, you have just pure UV light, and under those is with royal blue lighting. So the urchins themselves produce a diffuse green fluorescence, which can have some variation between the individuals. So speaking of external biofluorescence, not only did the urchins have a diffuse green biofluorescence in the spines, but they were also producing under stress conditions a quite significant amount of red fluorescence exudate. And you can see there in the pseudo RGB image on the left, you can see it quite clearly. And then also there were two broken spines that were fluorescing in a very high green pattern, that, as you can see in the graphs on the right. So one of the interesting stories that we've seen here through our preliminary analysis is that the external fluorescence of the animals has a general downward trend, while the internal coelomic fluid fluorescence has a general upward trend in the same period of time. My hypothesis is that this is in response to the stress, and with such a stress, you have a change in the excuse me, can you go outside? Yeah. Apologies for that. But so the hypothesis is that the fluorescence creates more of a disturbance. Excuse me. The external fluorescence decreases, and then the coelomic fluid increases its fluorescence. And what happens is that in my opinion, the red fluorescence is a byproduct of that stress and is exuded from the animal. So the coelomic fluid trials that we did on yesterday evening was with those three different groups, and as you can see, under 3 hours, the hot water animals had quite a large spike at around 680 nanometers, and followed by the out of water animals as well, but as not as a significant manner. But what's interesting too is that in the green zone, you have from about 495 to 590, give or take. The green, as time goes on, actually collapses in relation to the amount of red fluorescence produced by the animals. Now, for sea lice, concept was just published by a different firm in Norway at the end of October 2022. But as you can see, under normal and excitation lighting, sea lice are highly fluorescent, and I will be working with the team here in Nofima to produce a fluorescence guide, for species, sex, and age of the animals to help in the identification of them in aquaculture. This can be quite useful, in my opinion, because you can actually see these animals fluoresce while seagoing adult salmon do not fluoresce from their skin. So it would be quite useful tool for AI to pick up in their census or in their control efforts. Seaweed, we looked at 5 different species. On the top here in regular light, you can see the Zacharina, middle is the Palmaria, and the right is digitata. And under the excitation below, it's not a surprise, as chlorophyll is known for quite a while to be biofluorescent. But what is interesting here is that in the Palmaria in the center and in the digitata on the right, the animals, or excuse me, the macroalgae is producing some type of yellow exudate. What it is, I'm not certain, but it's something that the seaweed specialist here, Xinjin Wang, has not seen before either. So what it was interesting finding was that the different species have different spectral signatures. So the digitata was the highest, and with the saccharina second, and then the weedy species as a secondary species, the sargassum and the fucus being quite similar, and the palmaria being the least intense of the species. Now, what this can also be used for was reproductive changes between the fronds. Non-reproductive fronds were found to have a taller peak at around 680, while the reproductive fronds had a smaller peak at the same level with a secondary peak earlier on around 674. So what this could be used as is a way to identify fronds in aquaculture production that are going to be ready for producing I believe they're called gametophytes, the sori, before they're visually visible, excuse me, itself on the fronds. So I'd like to just say thank you to Phil James for providing this opportunity, Tina Thesland, Xinjin Wang, Samuel Ortega, and Tor Evanson in particular, and my mentor, Stein Cato Lindberg, quite possibly the smartest man I've met. And many a times I felt like Fry in that photo, when he's speaking to me about physics. I'd also like to thank Evan Derland with the NOFEMA's Deep Vision project for providing additional support for the investigation of aquaculture species. So thank you.
Thank you very much, Thomas. Biofluorescence is a very interesting topic, but why do echinoderms biofluorescence? Like, what is there no purpose to it, or?
Well, this is all. It's a good question because it is completely a novel. It's completely novel. So I haven't seen any published data on biofluorescent echinoderms. I suspect that it's similar to in purpose as bioluminescence, which is produced, I believe, by the luciferase enzyme. So many deep sea echinoderms are bioluminescent, but not biofluorescent. So I feel that this could be something for communication, and if not with each other, then as perhaps as a startle mechanism for predators or a signal for their predators.
Yeah. This is time for the Q&A session, and we all have a question from Marcel. Marcel, do you want to ask a question?
Put my camera on. So thank you for this nice talk. I have several remarks and a question. The first remark is you talk about biofluorescence. And biofluorescence is a very bad word, because David Gruber invented this word by imitating bioluminescence. For bioluminescence, both are my study fields. Bioluminescence, you need a living animal. You take a dead animal, it will never light up. However, for biofluorescence, there is no use to be living. It's just a pure physical phenomenon. You take a dead animal, you put a blue light on it, you will get the fluorescence until the animal starts decomposing. So a much better word for biofluorescence would be natural fluorescence. That was just a remark. With Jérôme Malfait, who is also bioluminescence, we are worrying against this stupid word biofluorescence. It's just not a good word. You do not need life to get biofluorescence, to get natural fluorescence. That was my first remark. Then a second remark was, it's more a warning. You say, for instance, that the lumpfish, the tupicles, they fluoresce you say the same with the spines of the sea urchins. And then I can just say, because I have often had the experience, that it could be another phenomenon than fluorescence. And that you can find out by exciting with other wavelengths. For instance, when you take a rainworm, you put a blue light on it, you get greenish fluorescence in all the cage. When you put a green light on it, you get it a bit orangey. You put an orange light on it, you get a bit reddish. So it's more a light transmission phenomenon. Mm-hmm. So I should say, take care by saying it's fluorescence. Verify whether what you are seeing is fluorescence. And the last remark I had, you said for the sea urchins, we found in the Northwest Passage, we found one sea urchin in which the small feet they have fluorescence nicely. So in that case, we are sure it's a real fluorescence. In the case of other sea urchins, some we had, the spines were lighting up, other spines didn't light up. But then again, I was afraid that I was not looking at fluorescence, but more on this strange light transport phenomenon, which is a bit like a light fiber. So take care by saying that it's fluorescence.
Okay.
But it's a very good idea to use to use natural fluorescence for trying to find out what's happening in those animals.
Yeah, and that's I'm working with Stein Cato, who's a great physicist, so I always double check everything, and I'm always on the like skeptical side, as he knows very well. That I always want to make sure that the data is there to, preliminarily, this is but it needs to be further evaluated and tested. In terms of using the natural fluorescence for stress, it seems to be quite promising in this, but again, it needs to be further tested and evaluated. But I appreciate your input and experiences, because it is
another remark. As you're rich and you have a hyperspectral camera, did you try to excite at longer wavelengths? Because you limit yourself to the UV blue, deep blue, but going higher up, you get other fluorescences perhaps. And as you have the camera to measure it.
Yeah, we did in our experiment, just to minimize the level of stress that the animals experienced too. We had a conveyor belt system, but we wanted to limit the time of the animals under scanning to a minimum. But we did we did UV, we did royal blue, and then we did regular light. So, and again, the amount of data we have, but that at this moment is massive. But I think it's a good idea to always double check at another light spectrum as well, to play devil's advocate.
Perfect.
Fantastic. Thanks.
Thank you.
Are there any other questions? I don't see anything in the chat. If you have a question, just raise your hand. I do not see any questions, so I'm just going to ask you another one. Oh, you have one from Philip James.
Hi, Thomas. Very nice talk. Thank you. I'm very familiar with the work you've done, not so familiar with the topic, but I guess what's most interesting to us in terms of aquaculture application, as you've described, is sure the biofluorescence itself, but the changes in biofluorescence that occur over time. You got any thoughts on further application of that in whatever species there might be in terms of aquaculture?
I think in terms of, again, biofluorescence is quite pervasive, or natural fluorescence. It's pervasive in the natural world, but again, not all species natural fluoresce, in my opinion, at least from preliminary evaluations. Salmon do not, for example. But in terms of applications, I feel that it could work for larval stage, scanning, especially for rearing of echinoderms, which should be investigated, and then looking for perhaps changes in bacterial growth in that otherwise is difficult to see on even fish and echinoderms during production, to see, especially that with like the European Union's mandate on reducing the amount of antibiotics used in production, early detection, I think is a key element and possibly good pathway for further research for this. So I feel that if you work through it and do some rigorous testing, there's many different applications for this if the animals show that type of natural fluorescence. But even with something that doesn't fluoresce, like salmon, their parasites seemingly do, and that can work for the managers and the veterinarians and whoever else on the team's favor to help have another important diagnostics like tool set in their kit of parts while they're working.
Yep, thanks. I can certainly say for urchins, we've been searching for a very long time for a non-invasive form of some kind of stress measurement. So I think the results you've got are really promising, yeah, and interesting.
Yeah. Thank you. Great.
We do have time for one more question. If anybody want to ask Thomas something, I will just give you just a few seconds to raise your hand or to write in the chat. I will conclude that no more questions. And I just want to thank you, Thomas, for your webinar today. And thank you to Marcel and Phil for your questions. And thank you everybody for participating. I posted a link in the chat for our webpage. And don't forget to follow us on Facebook, Twitter, Instagram, LinkedIn, and of course YouTube, where you can find a recording of this video as well as our previous webinars if you are interested. Thank you again, Thomas.
Well, thank you. So thanks very much.